All right, so today we're going to start off with a discussion on how organisms colonize the land. We went from bare rock to life pretty quickly. This is all about Chapter 26 and 30.1 of Biology in Focus. I love how it's 30.1 and that's it. It's like, don't worry about the rest, just that little bit. All right, so before we really get going, I want to start talking a bit about evolution. We've talked about evolution a lot. Like, that's our thing in this class. It's like evolution, we began with it. We talked population genetics and evolution, how these organisms derived from each other. So here's the deal with plants and fungus. They all started in the water. We've all come from the water in the beginning. Heck, you're still mainly made up of water. There you go. So plants and fungus started off as single cells living in the water. The difficult part about moving onto land for plants, for fungus, for animals, for anything moving from a marine environment to a dry environment is desiccation, or losing water. Now in the ocean, that was never an issue, because there's water all over the place. It was really easy to get water. On land, it's an entirely new environment. So they had to adapt in order to survive dry land. One way that they adapted was through the creation of 3D tissues. So a single cell washes up on the beach. It's going to dry out pretty quick. But if a clump of cells wash up on the beach, that interior cell will be wet or can stay wet for longer. It's sort of like a washcloth. You guys have gotten a washcloth wet at some point. You've taken a shower. Oh, I don't know. Maybe you don't use washcloths anymore. He's like loofahs. But the idea is, think of a washcloth. Think of it being very wet. If you lay it out if you lay it out flat, it dries out very quickly. But if you ball it up, it stays wet. The inside stays wet. How many of you guys have? You really needed some clothes, and you threw whatever was in the washer in the dryer, and you just hoped they would get dry quick. You know what I'm talking about? That moment of, I need a pair of pants, and throw it in. It balls up. You stick your hand in there after 20 minutes because you're like, I have to go now. You feel it. The outside is dry. You pull it out, and you're like, oh, good, I can leave. You shake it out, you start to put it on, and then there's that wet feeling. Yes, you guys know the clingy, terrible feeling. It's because the inside didn't dry out. The same thing is happening with three-dimensional tissues. The outside can get dry, but the inside will stay moist. Eventually, those exterior cells started to derive energy and derive moisture from the interior cells. There are a bunch of other adaptations. We'll talk about them later in the lecture. But what do plants have on the outside of them that repels water? Or even more important, keeps water on the inside? It's a macromolecule they secrete. Okay, back to 101. There are 4 kinds of macromolecules. What are they? Proteins, lipids, carbohydrates, and the one that we talked about an insane amount at the very beginning of this class, nucleic acids. Which of those is going to repel water most often? Lipids. Specifically, what kind of lipid is on the outside of a plant that you, even did a whole lab about? Wax. Wax is a lipid secreted by plants to retain water. So you've got three-dimensional tissues. You've got wax on the outside. So plants and fungus started off as unicellular organisms. They started to colonize the land, and they had to adapt to the land. So we're going to talk about them separately. First, I want to talk about fungus. Fungi are monophyletic. Oh, thank God, finally, something makes sense. All of the fungus that we all consider to be fungus are classified with a single common ancestor and all of the descendants. This is the one time taxonomy works. And you wouldn't know it by looking at them, but fungus are really closely related to animals. People always look at fungus and think they're like plants. They're not. They're really, really similar to us. Both animals and fungus are heterotrophic. Plants are autotrophic. Both animals and fungus undergo absorptive nutrition. They have to pull nutrients across their membrane. And both animals and fungus store their energy as glycogen. So animals and fungus are really, really closely related. And we saw that at the end of the last lecture where we were looking at nuclearia and the choanoflagellates. Very similar organisms. Okay, so this is Toad from the Super Mario Brothers series, in case you didn't know. And if you didn't know, you've been under a rock for the past 30 years. And that's weird because most of you aren't even 30. So here's what I want to know. Is Toad a fungus or an animal? Animal? Why? The mushroom's a hat, so he's an animal that just wears a mushroom on his head? He's got like a symbiotic relationship with it? That's how it works? Because I distinctly remember it's called the Mushroom Kingdom, right? Why is it called the Mushroom Kingdom? Not trying to trick you. Because they're a really big mushroom. So I guess this is then an animal who happens to live in a symbiotic relationship with fungus. I don't know. Maybe his hat's getting something off of him.
Why else would we always be so dang happy?
That's true. Why else is he happy? He's got a hat to lick. So life's good for them. That's why they need Mario to do everything. You go do that. We'll just be over here watching. Now, animals and fungus had a common ancestor, and it wasn't like Toad or his people. The common ancestor was probably an amoeboid protist. It's got these pseudopods coming out of it. It would eat algae, probably ate some bacteria through phagocytosis. What's phagocytosis? Right, engulfing another cell eating another cell. So these early amoeboids, they would eat by surrounding other organisms and taking them into their body and digesting them with their lysosomes. Once we get this group called nuclearia, you get this, so this amoeboid form, you get this new type of group called the Cryptomycota. They live in soil, and they lack a cell wall. The cell wall, or lack thereof, is important because this organism can continue to eat through phagocytosis. Once you get a cell wall, you cannot undergo phagocytosis anymore. Why? In case you forget, phagocytosis, engulfing something and bringing it in. It's not about protection. You'd have to break through that wall. Or you'd have to, every time you wanted to expand, keep building walls around it. And then you'd have to destroy walls and build more walls. It's like a horrible game of Civilization. So, aside from Cryptomycota, every other true fungus has a cell wall, which means they can no longer eat through phagocytosis. So, cell wall's good. You're not going to burst anymore due to osmotic pressure. Water can't flood into it, causing you to rupture. That's awesome. Downside is you need an entirely new way of getting nutrients. So here's where we get what's called the osmotroph. New type of eating, osmotrophy. You absorb organic materials directly across the membrane. You are an osmotroph. Fungus are osmotrophs. There's no phagocytosis, it's just direct diffusion. The way a fungus works is it's going to secrete enzymes on the ends of its, we didn't say what it's called yet. Okay, so on the edges of the cell, enzymes are going to be released. And what do enzymes do? In general, what do enzymes do? They break things down. They speed up reactions. They make things happen faster that otherwise wouldn't happen. So what these enzymes do is they break down these larger substances on the outside of the fungus and allow them to be dissolved across the membrane and across the wall. So it'd be sort of like if you were eating Nobody has food this morning. That's so weird. You got okay, you got your whole breakfast back there. Thanks. Holy cow, you've got a whole box of Dunkin' Donuts?
Wait, what?
No, that's awesome. I would totally do that. I'm going to do that to you guys. Your exam, I'm going to bring in a box of donuts, and I'm going to eat them all. Like, staring at you while you take your exam for 3 hours. So it'd be sort of, okay, I'm going to use your calculator. Sort of like if you were to eat this calculator. I don't recommend eating calculators. But if you were, you'd go over, lay your hand on it, digestive enzymes would come out of your hand, and then it would slowly be pulled up into your body. Doesn't that sound weird? Cool thing is that's what actually happens in your body. You have these intestines. We're going to see them next week. Not yours. Not yours. We're going to see it pigs. If we saw yours, that would be bad. So your intestines loop back, your small intestine loops back and forth and back and forth. Why does it have all the twists and turns? Surface area. You guys are going to just walk away. Anytime you hear twist and turn, think surface area. You have this increased surface area so that more cells can touch that undigested food going past you, through the small intestine. And as they're going through, the nutrients are being absorbed into the cells of the intestine. But wait, and then from there into the bloodstream. But there's even more, because even though you have these twists and turns in your intestine, you've got these thing called villi in your intestines that stick up like fingers. That increases the surface area even more, because not only are there twists and turns, but the twists and turns now have twists and turns. And then on the edges of the villi, there's even more fingers. So it'd be like having fingers at the end of your fingers. Twist and turnception. So now you have twists and turns on your twists and turns on your twists and turns, increasing surface area dramatically. All so that as much material that has nutrients in it as it's passing through your intestines can touch those things, allowing for absorption. You digest things this way, and fungus digests things this way. So no more phagotrophy, but we still can get nutrients. It all comes down to form determines function. Fungus have some really efficient shapes. The body of the fungus, and we're talking about body as if it has a body, but it's called the mycelium, is absolutely functionally adapted to osmotrophy, to increasing surface area. It's made up of these things called hyphae. They are very long filamentous strands that grow insanely fast. The mycelium is the body. It's just a whole bunch of intertwined hyphae wrapped around each other, creating a network. Wherever these hyphae touch, they can start secreting enzymes to break down the material around them into nutrients. By far, the largest organism on Earth is a fungus. Everybody always assumes blue whale. That's the largest animal. Dinosaurs were pretty darn big. But fungi are huge. There's one in Oregon that takes up 2,200 acres. And assuming it didn't burn, it's still there. It's called the humongous fungus. Literally, that's what it's called, the humongous fungus. They know it's the same fungus because what they've done is they've taken a radio-labeled dye, a nutrient, and put it on one end of this range. And they were able to track it travel through the hyphae of the single fungus all the way to the edge of the range until they figured out this broad size. They're huge. And they live entirely underground. For now, until they erupt and take over. Now these hyphae can be termed septae or aseptae. All that means is septae is they've got cell walls separating the cells. Aseptae, the cell walls have disappeared. Because they can be septae or aseptae, there might be many, many, many nuclei in a single, what we would consider to be a single cell. Why do they need so many nuclei, so many nucleuses? Do you think? We've got these big, giant cells now. So why do you need 15, 20 nuclei in it? Yeah, that's exactly it. Because having just one nucleus, let's say you've got a control center on this side of the room and you want something to happen way, way, way over there. Well, you have to write out the plan and walk it over here, and then it gets dropped off and the plan happens. That's fine. That might be fast enough, but let's say then the signal needs to go all the way to the other side of campus. It's going to take time. So to diminish the amount of time necessary, you have sort of substations set up to pass on plans. Now you can have this happen if you have many rounds of mitosis. Remember, mitosis is the division of the nucleus without the coupled cytokinesis. So the cell never splits, just the nucleus. So what's the point, and you guys should get this pretty quick, of having such really long, thin hyphae? Surface area. When in doubt, surface area. So fungus have to eat. And the way they eat is osmotrophy. They got this cell wall in the way, so they have to digest stuff on the outside to bring it in. Yes? So osmotrophy is diffusion across the cell wall. To optimize osmotrophy, to get more nutrients, you have to have a huge surface area. Here we've got a fungus interacting with a plant. The roots are down here of the plant. The fungus are spreading out from the roots. This fungus is not eating the plant. It's actually acting in symbiosis with it. The plant is providing it food, and the fungus is actually giving the plant nutrients. So by having lots and lots of long, thin hyphae, you're able to increase the ratio of surface area to the internal space. It's the same reason that Arctic animals are big and fat. They want to minimize the amount of surface area to internal space. Whales are huge. They can survive in very cold weather because their surface area compared to their internal space is minimized. They have a lot less surface area compared to their guts. And that keeps their core body temperature warmer. It's called thermal giganticism. Fungus are working in the opposite regard. They want to be super long, super thin, so they can touch everything. If you were to take a fungus and put it into a nutrient-rich environment, the hyphae would grow and grow and grow very, very quickly. You guys have seen, this happens to you guys. It happens to me all the time. You go and you get bread. You're like, I'm going to eat this bread. Fresh bread. A day or two later, covered in mold. Been there? Don't you hate that? Because then you feel like I just wasted my money on bread. Doesn't happen with the bread that has preservatives, but if you get fresh bread, it happens pretty quickly. Under optimal conditions, moist, warm temperature, lots of nutrients, hyphae can grow at a rate of 1 kilometer per day, 2.5 feet per minute. Okay, that's really, really fast. That's something you could see if you were watching a single microscopic hyphae extend. The way they grow so quickly is they use osmosis to their advantage. So water is going to flood into the hyphae from the sides. That's creating pressure. The pressure wants to go somewhere. So the Golgi apparatus is shipping out nutrients toward one edge. Nutrients are coming, cell wall components are moving this way. Water is pushing them forward. So you're using pressure to sort of like push. You guys have been in a line, right? And there's somebody in front of you who's not going quickly. Oh, you do this at stoplights. You see somebody that should be going, and you're like, I'm just going to start creeping up on them. Just a little bit. So just let them know that it's time for them to leave now. Yeah, so it's the idea that if you got pressure behind somebody, they tend to move forward. Oh, grocery lines. Just, push your cart a little bit forward a little bit more. Maybe they'll get the hint. You put your stuff right behind there. So just, come on, let's go. That's what's happening here. Water's pushing in this direction. The pressure is able to extend the cell wall. Cell wall components are being put there. But in order for that to happen, you've got to have space over here for it to grow. Well, that works out because enzymes are constantly being secreted out the front. So there's an open space here, nutrients being brought in, and the cell is growing through the space it just digested. The Golgi apparatus is responsible for shipping components out of the cell. So you're going to take a polypeptide from the endoplasmic reticulum, get it to the Golgi apparatus, where it's then shipped out. That enzyme starts to break things down on the outside of the cell, and that process is through exocytosis. So as the fungus grows, it breaks down whatever it's eating, fills the space that thing it was eating was in, and then starts to digest more around it, and keeps growing and growing and growing. So, you guys have seen cheese go bad before, right? Mold all over your cheese. It happens all the time in my refrigerator. I forget I have cheese, and then suddenly it's growing. There's this idea that you can just, don't worry, just cut off the bad part. Has anybody ever heard that? Yeah, it's okay, just cut off the bad part. The fun part is, if you think about it, you know that hyphae grow at about 2.5 feet per minute. If you cut off the bad part, all you're doing is just convincing yourself that you're not actually eating fungus. Fungus reproduce through asexual reproduction. One way is they reproduce through asexual reproduction. The tips of the hyphae can produce asexual spores, and those spores are called candidia. They will bud off of each other. So you got the tips of the hyphae, they produce these little tiny baby spores, and those spores sort of just pop off, and they float free. After that candidia land on a substrate, after about 2 days, a single fungal spore can produce 40 million more candidia per hour. Have you guys ever seen, it's one of those moments, you look at out the window and a sunbeam is coming through. You know what I'm talking about? Like cats like to sit in that sunbeam. Have you ever seen the little things floating in there? And you're like, huh, there's stuff floating there. Likely, a lot of the stuff you're seeing are candidial spores, dead skin, and fungus. The more you know. Candidia looks and feels like dust. It's very, very easily spread by touch. It's easily spread through the air. If these carpets have not been washed, and I can guarantee you they probably haven't in the past, several months, they are filled with fungal spores. Candidia is pretty common. You see it all the time at the store, right? Citrus mold. The feel of citrus mold. This is a high concentration of candidia. So once you see a bag that's got those fungal spores in it, don't get that bag. It's pretty much on everything. But wait, there's more. If you see these fungal spores, this high concentration candidia, in the middle of a pile of good lemons, what does that tell you about the other lemons? They probably have candidia on them, because you know there's a lot of air motion, there's a lot of touch going on. So, giving you a heads up on that. That's just a common example. Another form of reproduction is budding. This is when a daughter cell forms on the surface of a larger cell and just sort of pops off. It's almost like, I don't know, if you had a pimple and it grew bigger and bigger and bigger, and then it popped off and became a little you. Some organisms reproduce, fungus reproduce through budding. Some common examples are infectious yeast. A yeast infection will reproduce through budding, and athlete's foot. So what's happening with athlete's foot, if you've ever seen somebody with it, is there's a fungus. And that fungus, the bottom of your skin, the bottom of your feet are dead skin. Hey, anybody ever gotten a pedicure before? Isn't it amazing how much skin they can take off with that? And it doesn't hurt or anything. Well, that athlete's foot, that fungus is eating the dead skin underneath the layers. So what happens is, as it's eating, sections of the skin are flaking off because they no longer have a substrate to bind to. And fungus reproduce through sexual reproduction. Sexual reproduction increases genetic diversity, allowing them to survive environments they otherwise wouldn't. Hyphae are going to be integral to sexual reproduction. So if you have two hyphae of different sexes, so boy hyphae, girl hyphae, they come together, they meet. And where they meet, they start wrapping around each other like this, and they start to form what's called a fruiting body. Now, some fungal species can have up to 36,000 sexes. Okay, humans, how many sexes do we have? That's a loaded question, right? So on the conservative end, we've got 2. On the upwards end, you might have, 18 or so. Fungus, though, even if you're the most complicated human out there, doesn't compare to fungus. 36,000. That's the size of a small town. Imagine trying to find a mate where there are 36,000 different genders and you don't know which one you're compatible with. It's a lot of experimenting. And fungus do a lot of experimenting. The way they figure out who to mate with is not the same way you do. What they end up doing is they reach out to everybody. They're kind of gregarious that way. They're like, hey, can we mate? Touch my hyphae. Came out totally wrong. Sorry about that. If the hyphae are able to secrete compatible enzymes, compatible proteins, they begin to wrap around each other. If they don't, well, suddenly you've got a competitor who you can't mate with. We've got to wipe them out. So mate or fight. That's pretty much the option between fungus. When they start to wrap around each other, then you get an above-ground structure getting formed. So again, the only way you can determine gender, the only way a fungus can determine if it can mate with another fungus is by accepting a certain protein. Some fungus have certain protein receptors on their outsides. Others are able to secrete the protein that stimulates them to undergo these behaviors. So probably what you think of as a fungus, the mushroom, is really just the fruiting body. That's where the hyphae are twisting up among each other. That's where the fungus are having sex. Underground, that's where the fungus lives. This is where they go to mate. And by go to mate, I really mean those hyphae come up, intertwine, and you get that structure formed. Its whole job is to produce spores. So why would it be better for this why would they not mate underground? Why do they mate up high? What does that do for them? Right, so it produces spores and then the spores rain down. So it's able to spread spores farther than underground they'd be able to. So when you think about it, you guys have gone and kicked mushrooms before. I assume that was a pastime as a kid. Mushroom growing, kick. I mean, we've all done it. So when you're doing that, you're helping them release their sex cells. You're just, well, you killed it too. Well done. But you've knocked the spores far away from the parent's body, so now that can take root and grow as a bigger, another fungus. Do you guys know these things, puffballs? You totally did those, right? You saw a puffball and you're like, oh, I'm jumping on it. I'm totally jumping on it. And you jump on it and a big cloud of stuff comes out. That cloud that was released are sex cells. So what you just did was you helped the fungus release its sex cells. Don't think too hard about that. Fungus have been around to help human civilization, or humans have dealt with fungus for our entire cultural history. All the way back to the beginning of time, we have interactions with fungus. Beer. Beer was important. Beer is still kind of important, but beer was really, really important. We use yeast, a type of fungus, to produce the ethanol in beer. Now, today's world, beer is not used for what it used to be used for. Today's world, we drink beer in order to get intoxicated. Previously, we would drink beer because it was safer to drink than water. Water's got a bunch of horrible, horrible things in it, especially when we don't know much about sewage. People go around defecating near water supplies. Not now as much. They used to. There were all sorts of parasites in water. You drink water, you can get something. But beer, You brew something up with yeast, even a very weak beer, that will kill the parasites. So what people would do was they would drink beer, the watered down beer instead of water. That's the way it worked. Historical fact, there were elephants in England, and it happened that Henry I of England was given an elephant, and I believe it was by the King of France. There's actual records of this. Rather than giving the elephant water from the heavily polluted Thames, because everybody knew, don't drink water from the Thames. People literally threw out their feces every morning into the street, and then it got washed down to the Thames. Don't drink water from there. So they said, this is an amazing gift, this large elephant. We do not want it to die, so we will give it beer. Again, receipts and records from the time frame show that this elephant was given 12 gallons of beer per day. And it lived in, at the time, the, what's it called? The Tower of London was a menagerie. It was a zoo. There were polar bears there and a drunk elephant. How amazing is that? It's like, look, we got this. This needs to be safe. We got to have a drunk elephant. I believe the elephant ended up dying of liver damage. Would the elephant? Probably not. And I assume the elephant would probably have also developed a tolerance to alcohol after 12 gallons a day every day for its whole life. Alcoholism, right? Another thing that's really important that we use yeast for, that we use fungus for, is bread. Before we started using fungus in our bread wouldn't rise. It wouldn't last as long. It was a paste and it was flat. We started to incorporate yeast into it, and that made a harder bread that could last longer, which meant we could travel with it or store it. There's some downsides to fungus. As we alluded to with toad and being happy, ergot is a fungus that secretes what's called an aflatoxin. This has been known to have psychedelic effects, cause hallucinations. Now, that's one thing if you're meaning to have those, but if you accidentally have them, it's kind of freaky, right? Well, you don't know. But the idea is, during the Salem witch trials, people went nuts. They started accusing all of these older women of witchcraft left and right. It was mass hysteria. And what we found is the conditions, the weather conditions of the time were perfect for ergot infesting wheat. And we've actually there's been archaeological digs where they found ergot spores in wheat of the period. So what they think happens is whenever they had these big batches of witch trials, it was because people were hallucinating. Spiders crawling in their skin. Seeing things that weren't there, and they had to blame somebody. So they said they had a spell cast on them, when really they were just super high and really tripping. Probably the earliest form of fungal use that we've documented is the Egyptians using it to kill insects. And you probably hear people, you sprinkle a little bit of fungal spores onto an ant colony or an ant trail. They take it back to the queen eats it, and the colony dies. This is how they controlled pests in the pyramids. Animals use fungus. They use it for their own good. Leafcutter ants. What do you think they do? They cut leaves, right? Because they're called leafcutter ants. But they don't eat the leaves. They cut leaves, they take it back to their colony, and they drop it into a big pile where they're growing fungus. And they eat the fungus that they're growing. They're little ant farmers. In the same vein, animals do have a relationship with fungus. One reason, you guys know bees are sort of disappearing now, right? They're not totally endangered yet, but they're disappearing. One reason that bees may be disappearing is because of a mite that's carrying a fungus that's destroying the hives. Fungus partner with not just animals, with leafcutter ants and things, but they also partner with plants. And that kind of is weird because we always think of fungus eating plants. But fungus and plants develop mutually beneficial relationships. You could have a relationship between the hyphae of the fungus and the roots of the plants. And this is called, these are called endomycorrhizae and ectomycorrhizae. What does endo mean? In. So ecto is out. So what's happening is these fungus have a different relationship with the cells. Endomycorrhizae are going to invade the cell membrane. I'm sorry, the cell wall. They're going to be on the inside of the cell wall. They live between the cell wall and the cell membrane. So they can deliver nutrients directly to this plant cell, and the plant cell can give the fungus sugar. Because hyphae grow so much faster and are so much longer and more efficient than roots, this allows the plant to get a lot more nutrients than they otherwise would have. So the plant grows even better. Ectomycorrhizae, on the other hand, are on the outside of the cell wall. So ecto, outside. But they do the same thing. They bring the plant nutrients, and the plant gives it nutrients in return. Not only do you have hyphae interacting with the roots, you might have fungus living inside the plant. These are called endophytes. Endophyte technology is sort of on the, I guess they call it the bleeding edge. The idea is you're going to have fungus living inside of the tissues and producing something for the plants. They might secrete toxins. So you have a fungus inside the plant. The plant is giving the fungus nutrients. The fungus is in turn producing a poison that doesn't affect the plant, but will kill or harm animals that eat the plant. It might encourage uptake of materials. It might allow the plants to grow even better. So this experiment shows endophytes and no endophyte in dry conditions. The endophytes help take up the material better. Then we have lichens. You guys have seen these growing on trees before, right? See if there's a tree out there. There's a tree right outside. Does it have lichen growing on it? No. Okay. Lichen, they look sort of like green flakes. They're green, they're photosynthetic, but what a lichen is it's not a fungus and it's not a plant. It's a symbiotic relationship between algae and fungus. The algae is making food and oxygen. It's making glucose, it's making oxygen. The fungus, in turn, is eating the glucose, breaking it down into carbon dioxide and water, which the plant needs. Lichens are usually the first organism to take hold on bare rock. They're the things that have to come around first in order to create soil. Lichens are important for three big things: food source, soil building, and air monitoring. And it's all about reindeer. In poor weather conditions, if it's really, really cold, the lichen survive when everything else dies. Reindeer go rooting for lichen. There's not plants around. There's no bushes around. It's one of their favorite things to eat. That's why they kick around snow, so they can eat the lichen. Gives them a food source. Christmas at my house is really fun. Everyone's like, quick, give the reindeer a carrot. And I'm like, kids, go get some lichen. Put it on a plate. You get better presents that way. Lichens are going to secrete an acid that breaks down rocks and will slowly break down plants. That will allow for dissolved nitrogen to be released into the soil. So rocks are going to have all sorts of atomic components to them. We have to get those components from the rocks to the soil. These are nutrients that wouldn't otherwise be made available. And they monitor the air. Sulfur dioxide, it's a really, really major air pollutant. When sulfur dioxide is high, lichen dies. If you don't see lichen around, there's a real problem with air quality, you should probably not be in that area. So there we go. That's my fungus lecture. Now, fungus are closely related to plants. And this is my, and I had to even write segue. It's like, here I'm trying to segue. I'm trying to connect these two lectures. Algae are considered to be both a protist and a plant. They're connected to fungus through lichens. So fungus and algae work together to create a lichen. And those algae are the simplest of the plants, the very, very simplest. Plants. Plants have 4 properties. All plants have 4. That's right, 4. 4. 1, 2, 3, 4. 4 properties. You're feeling 5? We'll make up a 5th. Plants all contain plastids. They're multicellular. They have tissue, and they have a specialized reproductive system. So all plants, all the organisms that we know of as plants, everything that we call a plant has plastids. Do you guys know what a plastid is? Ever heard of a plastid? Okay. One of the organelles that we know of has the word plast in it. What is that? The chloroplast. And the chloroplast, the main plastid in plants, produces something. All plastids produce an organic molecules. Now that I think about it, we have talked about plastids because we talked about plastids when we talked about prokaryotes and when we talked about protozoans. All true plants are multicellular. That means they have lots and lots of cells that are connected together. They have cellular adhesion genes and cellular communication genes. Oh, there's the organelle. So plastids produce, they convert inorganic materials into organic materials. What's that plastid? The chloroplast. Hey, why does it have a whole bunch of twists and turns in it? That's right, surface area. Well, why do things have twists and turns? Eventually, that's a big concept. So one of the large concepts for Bio 102 is not just form determines function, but this idea, actually, it's exactly it. Form determines function. All of these motifs, twists and turns, providing more surface area, are going to come up again and again and again across the board in all organisms. So, is that a plant? It's a mushroom, and that's a fungus. How about that? Is that a plant? No. Why? That's right. It's unicellular. Is that a plant? Yes. In case you didn't know, that is a plant. Is that a plant? Nope. Unicellular. Over there. Oh, what's that? Is it a plant? No. How about that? Moss. Yes. Moss is a plant. Algae up there in the corner with kids swimming in it. No. Algae? No. How about, which one was that? Nope, unicellular. Oh, kelp. Kelp? Maybe. Could be. It's an algae, that's for sure. It's a whole bunch of unicellular things holding on to each other, but they're acting multicellular. So that's one where they're kind of iffy. Oh, cactus. Yes. Plants have tissues. Tissues are close associations of cells of the same type. In lab, you looked at a lot of cell tissues. The reason that there's tissues, the reason these cells are multicellular is all about retaining water. You have to live in the terrestrial environment. So you have some cells that secrete wax, other cells that prevent water from escaping but let gases in, and then yet more cells to conduct photosynthesis. Plants have some tissue that's called meristematic tissue. That meristematic tissue is where you're going to have lots and lots and lots of mitosis. Lots of rapid cellular division. That's where the plant actually grows. Anybody ever grown tomatoes before? 1, 2 people. Okay, good. 3, thanks. You feeling 5? I'm feeling. Anyways, so what happens when you're growing a tomato plant is it starts to grow upwards. The apical meristem, it goes up. It also grows off to the side. So you're going to start seeing shoots coming off the side. And right at these nodes, you're going to get another shoot growing. So you have axillary meristem and apical meristem. For those of you who have grown tomatoes, a deer comes along and they eat the top, which they inevitably do. What happens? Does the plant keep growing tall? No, it can't. It can only grow sideways now and out. Certain regions of the plant grow very, very, very fast. Up near that meristematic tissue, down at the root caps, down at the bottom. The rest follows. The way I think of it is, have you guys ever seen snakes at, not real snakes, these fireworks snakes where you light them and then there's a whole bunch of ash that comes out and starts doing loops? If you know what I'm talking about, you've got the meristematic tissue on one end. The rest just sort of grows out of that. This is where all the growth is, and the rest is just elongation. Now, it still comes back to these plants were adapted to a marine environment. It all comes back to water. Reproduction on land is based on the idea that these organisms are no longer in a marine environment. They no longer survive there. They need water. In order to rapidly colonize the land. Oh, you guys always want to know what the essay question is. This is one of them. Alternation of generations. Quick, well, Matt's not here. Alternation of generations is the plant's reproductive cycle. And what we're going to do is alternate between gametophyte and sporophyte stages, between an end stage and a 2-end stage for their number of chromosomes. So it starts off, you can start off, I guess, anywhere. Let's start off where we're kind of comfortable. An egg and a sperm, haploids, end cell plus end cell. They come together and fuse to create a zygote. That zygote begins to undergo mitosis to become an embryo, and it grows big and tall into a 2N organism. So far, so good? That 2N organism produces these things called sporangia. And the sporangia, so still 2N, produce their spore mother cells. Another cell that's 2N. Those 2N cells produce a whole bunch of spores through meiosis. So this is where you're getting genetically distinct haploid daughter cells. Those N spores land, and they start to grow what's called a gametophyte. A gametophyte is made up of identical N cells, so identical haploid cells. Those spores have landed, and they start to create a new structure. That structure will produce either eggs or sperm, sometimes both, that are absolutely identical to each other. So for us, our eggs and our sperm are vastly different. But for plants, their eggs and sperm are going to be identical to a whole bunch of other ones produced in that gametophyte. Then we're right back to where we started. The egg and sperm combine, you get genetic diversity forming, genetic differentiation, zygote to an embryo to a sporophyte, to a sporangia to creating a spore mother. The spore mother undergoes meiosis to produce genetically distinct spores. Those genetically distinct spores land, produce these haploid structures, these gametophytes, that then produce eggs and sperm. It would be as if somehow you spilled some of your sex cells on the ground and they formed a whole new person that could produce more sex cells so you didn't have to. This gave early land plants a huge advantage when colonizing the land because they could produce a ton of gametes. That let them spread like crazy. So because of having that gametophyte and undergoing mitosis, which is less energy intensive than meiosis, you made lots and lots and lots and lots and lots of sex cells everywhere that fused to make plants all over the place. So this new environment of land got colonized. Alternation of generation encourages new structures to form, like sporangia, that will have very, very specific functions. Here you have a moss. And if you've ever looked at moss, there's some up in the lab, you'll see these shoots coming off of it. So you have the gametophyte down here and the sporophyte up there. This is going to produce spores that have to fall down. So just like with fungus, you have a really large, very high structure, and it will rain down spores. So here we have in moss the gametophytic structure and the sporophytic structure. The gametophyte is N. The sporophyte is 2N. The whole point of gametophytes is to produce haploid gametes. They are genetically identical. Hey, do you guys know what movie that's from? Just curious. What? You didn't know? Is your mouth full? What? It's from The Parent Trap. That's right. Hayley Mills' Parent Trap. These gametophytes are called gametangia. These are the structures that actually produce gametes. You might have an archegonium or an antheridium. Once you've got these gametophytic structures forming, some of the cells, they are all haploid, they're going to form gametes. Other cells form around the gamete to make a gamete jacket. So it's like having a little like a gamete and then like armor around the gamete. The structures that produce eggs are called the archegonium. They look like little vases. So again, you see haploid gamete inside, and these are also gametes on the outside producing these structures, not gametes, they're haploid cells producing structures. For males, you have the antheridia. So you've got the male gametophyte, the antheridium grow off of it, and they're going to produce sperm. So in case you didn't know how reproduction works, here's how it happens. When it's moist and the sperm is mature, the sperm is going to need to get from the antheridium to the archegonium. It'll then travel down the length of the archegonium where it will find the egg. There you can have fertilization. It still depends on water. The sperm is flagellated. It has to swim. It doesn't just fly there. So when you don't have water, plants cannot reproduce. In the spring, if there's not enough water, you're not going to get crop fertilization because the sperm needs water to produce a fruit. So the gametophytes. The sporophytic structure is a 2N structure. We've got these two gametes. They fuse together to make the zygote. That zygote is diploid, but the cells surrounding the zygote are haploid. That zygote starts to develop into an embryo. All land plants are termed embryophytes because they make embryos. Every single land plant you know of. That's because land plants have babies. These embryos develop over time, and they entirely depend upon the mother. The mother tree, or the mother plant, produces excess cells that are going to provide nutrition to the developing embryo in the form of the cotyledon. This is called matrotrophy because you're literally eating your mom. The mother is providing a lot of energy to these developing offspring. So I'm not going to go into the discussion. It's very important to nurture offspring. You have to make sure that those offspring have energy to get going. There's a relationship we're going to talk about when we get into ecology, where there's a trade-off between the number of offspring you have and the amount of energy you give that offspring, the amount of maternal care you give that offspring. Turns out the organisms that are able to devote a lot of energy to maternal care grow larger offspring, but they produce far less of them. Humans, we don't produce terribly many offspring in our lives, compared to plants anyway, compared to even puppies or cats, because we end up caring for our offspring a lot longer. So cells within the sporophyte undergo meiosis, and they're going to produce genetically varied haploid cells, and those haploid cells are called spores. So we're right back to alternation of generations. Glad you're back, Matt. This is important. So we're going to go from, let's see, we'll start at the gametes again. The eggs and sperm, they fuse to make the zygote. The zygote develops into the embryo. The embryo develops into the two-end sporophyte. The sporophyte undergoes meiosis to produce spores. Those spores land, undergo mitosis to produce haploid gametophytes, which then in turn produce haploid gametes, and we're back to the beginning of the cycle. We're alternating between diploid and haploid. On the sporophyte, the structures that produce spores are called sporangia, and they have lots of different shapes to them, and it all comes down to form determines function. Here's a sporangia. It's got a very, very thick wall to it. Why does it have such a thick wall? Well, it's because it's right over there. I was going to say, why does it have a thick wall? Well, it keeps out pathogens, bacteria, fungus, things that want to get inside and eat the sweet, delicious spores. It also stops ultraviolet radiation. Why would a plant want to stop ultraviolet radiation? What does ultraviolet radiation do? It does the same thing to plants that it does to you. It harms them. How? Yeah, ultraviolet. How? Yes, by breaking down the something that's very, very, very important to us. No, even more important. Keep going. Way more important. Some DNA. Ultraviolet radiation breaks down DNA. What it does is it causes these things called shoot, I don't teach a genetics class here, but I wish I could. There are these things called photodimers. Thymine photodimers. So you know what a thymine is, right? So if ultraviolet radiation hits DNA and you've got two thymines right next to each other, they fuse to each other. And now you've got this bend in the DNA so that next time it's copied, there's an error. And those errors build up, and that's how you end up with cancer. So if you go out and tan a lot, you're going to die a lot faster than the rest of us. Ultraviolet radiation can also hurt these plants for the same exact reason. So sporangia are modified in such a way that they can help disperse these spores. For instance, here's a bell. It's got these little fingers on it. So the spores go out through the fingers like it's sifting them. So instead of them just sort of dropping straight down, they get sifted out and spread a little bit wider. They can get caught in the wind. You might have bells. These are on the underside of ferns. So what happens is wind comes and it blows the fern, and these bells swing side to side. And if you've ever, taken a glass of water, I don't know, you're singing a drinking song or something, you're like, la, 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 and you're swinging it back and forth because that's how you sing drinking songs. I'm really tired. I think I told you guys that. Water splashes left and splashes right, just like these spores would splash. They disperse out far. So, if we talk about bryophytes, we can pull all this together. Bryophytes are mosses. They're a very, very, very simple plant. Here's an example. You've seen these growing everywhere. They grow in all sorts of places. Anywhere you have moisture, you're going to have moss growing. They don't need the same nutrients that other plants do. They can live outside of soil as long as they're pulling up water from their environment. Typically with moss, what you're looking at is the gametophyte. You don't see the sporophyte very often. But if you look really closely, sometimes you're going to see these higher structures that are the sporophyte sticking out. And again, they're higher so they can disperse spores farther. Mosses, though, can't grow tall. You can have trees that will grow really, really high, but you don't see moss that's like a foot deep. Why? Why can't moss grow tall? Because it all comes down to water. Mosses need to get water, any plant needs to get water from the bottom where the water is to the top. The problem is mosses can't take, they have to get water directly into their cells. They have to do through direct uptake. They can't suck it up like a straw. There are no straws in moss. So they take water from the environment directly into the cells instead of having some other cells deliver it to them. So you can only build a tower so tall of water balloons. You guys ever made water balloons? Have a bucket of them ever? You like make a big bucket of them? You can't keep filling more water balloons into that bucket or the ones on the bottom will burst. Same thing happens with moss. These things are, they're pretty much just water bags. They've got cell walls, but that cell wall is only so structurally sound. It cannot continue to support excessive weight. It's because mosses do not have vascular tissue. Vascular tissues provide both structure and support. They allow for the movement of water and nutrients. So typically when you look at a tree, what you're seeing is the sporophyte structure. Inside that sporophyte structure, bringing water from the bottom up, you're going to have vascular tissues. Giving it stability and strength, you'll have vascular tissues. Parts of the plant, and I have to go through this as if you've never seen a plant in your life. You have branches. So far, so good. Branches allow for plants to grow bigger and taller. They also allow for greater surface area. Why? You see them twisting and turning. Bigger amounts of surface area exposed to, that the leaves are exposed to. Yeah, click surface area. Surface area. They also possess, like I said, tracheids, which are very specialized cells that allow for the movement of water and nutrients, of which I'm concerned that you understand two of them, the xylem and the phloem. So over here, we've got the xylem. You'll notice they're called tracheids because they got these little tiny perforations, these little holes in them. Over here we have the phloem, same basic structure, but you'll notice that the phloem has these larger perforated plates in it. We're going to get back to those in just a minute because I want to talk about the tissues, stems, roots, and leaves. We're going to talk about the tracheids when we talk about the stems because stems are made up of xylem and phloem. They're the highways and byways of the plant. Phloem is primarily composed of tracheid cells, so it's got lots of holes in it, and that allows for the movement of nutrients and water through the plant. I know that was a question you guys had, and so there it is. It's two-way flow. It's not just one direction. It's got two directions to it, both up and down. Xylem is made up of lignin and really allows the plant to grow tall. Yes, you can move water and minerals, but you're moving it in one direction. The primary purpose of xylem is to act like a straw for both structure and to pull a bit of water up. How can I put this? All right, straws. You guys have used a straw before. Have you guys ever stuck a straw through an orange? Do you ever do that experiment? You take a straw, hit an orange, it goes through? Now you can. Next time you go to anywhere where you have an orange, take a straw, and you can easily stab it. In fact, you can kill a person with a straw. If you take a straw and go to their temple, right about there, the straw will penetrate. It's got that much strength. Sorry, I'm tired again. Little things you didn't know about me. The idea is a straw is stable and structurally sound in one direction. It can take a lot of vertical stresses. It does not take horizontal stresses. Look, I was a very angry graduate student. Roots are very specialized organs that take up water and nutrients from the soil. So they've also got tracheid tissue in them. You're pulling water up and into the rest of the plant. Often the roots form a mutualistic relationship with fungus. Fungus are going to increase the surface area of the plant. Leaves are on the edges of the branches. Leaves are flat. Why are they flat? When in doubt. Surface area. Flat leaves maximize the amount of surface area exposed to the sun. So we've got xylem and phloem. Phloem is moving food and water. The xylem is giving it structure and moving water in one direction. Vascular plants have to keep water on the inside. And again, in case you haven't noticed, I've talked about this, 3 times now? 4 times? Because it's an important concept. Probably another essay. The idea of water retaining adaptations in plants and fungus. These 3. These are, yes, some of them. 3D tissues, the other one. A waxy cuticle, a stoma, and internal conductive tissues. As we mentioned earlier, wax prevents water loss. Wax is hydrophobic. It repels water. That waxy cuticle stops water loss and stops pathogen attack. Whenever an insect has to bore into a plant, they've got to get through this waxy cuticle. If it's thick enough, an insect's mouth parts cannot pierce it. You'll have stoma. Stoma, I believe, we looked at in Bio 101, I think. They're complexes of cells that open and close to allow carbon dioxide into the plant. They let oxygen out of the plant. In the process, sometimes, though, they're also going to let water out with transpiration. And that's not good for the plant. If there weren't stoma, there would have to always be an opening to allow for gas exchange across that wax, that waxy barrier. By having stoma, you can open and close the door. It's sort of like the difference between having a door and not having a door with an air-conditioned house. You're constantly trying to keep the house cool, but if you leave the door open, all the cold air goes out. And it costs a lot more to keep the house cooled. In this case, the coolness is the water. Plants need to keep water on the insides. And then those 3D internal conductive tissues. So 1, we've got 3D tissues like we talked about at the beginning of the class. And 2, the xylem and the phloem are on the inside, not the outside, which means they're not leaking water to the outside. Water's not evaporating very quickly. That's it.